Okay, first question, we're looking for two purines. So purines, remember, I have a trick where pyrimidines, because it has a Y, those are our bases that have Y's in them, so that would be thymine and cytosine. So we're looking for a combination of two bases that are neither thymine or cytosine, which means A, adenine and guanine. Just as a second check there's two N's in adenine, two N's in guanine, which means that they should be double ring structures, so those are the correct diagrams of them. You might have thought BY isn't uracil, a purine, but it's replacing thymine, which we know is pyrimidine. Okay, question number 2, we're looking for statements that are not correct. Chop the kiwi fruit, that is a step that we do, but that's not to break open the cell membranes, that would just break the cell wall, so the correct answer there is A. Okay, question 3 was probably the first troublesome one. So which of the options contains the correct definitions for non-overlapping and degenerate? So each nucleotide is only part of one triplet of bases, that's correct for non-overlapping, but the molecule breaks down easy is not correct for degenerate. Gene follows straight after each other and the molecule breaks down easily, neither of those are correct. C, each nucleotide is only part of one triplet of bases, that is non-overlapping. More than one triplet codes for a specific amino acid, that's the definition of degenerate, so we're looking for answer C here. It's always worth reading through all the options. So genes follow straight after one another and more than one triplet codes for a specific amino acid. This is a problematic statement because actually some genes overlap with different reading frames, so D is not correct either. Question 4 is looking at DNA replication going from 3' to 5' end, so it might be useful to draw yourself a little nucleotide there to think this one through. This means the lagging strands have small gaps left in the backbone. DNA ligase works to seal these gaps. Which of the options A to D identifies the bond formed? So it's saying this bond here, that's the phosphodiester bond, so your answer there is B. Okay, in question 5 we need to read all these statements. So genetic code has redundancy and is degenerate. I think this term might have just confused people a little bit. Those two things mean pretty much the same thing. Okay. There's only one codon for the amino acid methionine, that's the start codon, so 1 and 2 are correct. Codons act as stop and start signals during transcription and translation. That is actually not correct because that stop and start is just for transcription, not for translation. Prokaryotes generally use the same genetic code as eukaryotes, that's correct. I mean, if someone wrote it in an exam, I'd take out the generally. mRNA codons have the same nucleotide sequence as DNA triplet codes. That is not true because we know that uracil replaces thymine, so incorrect. So it's 1, 2, and 4, which is option B. Okay, for this question, number 6, we wanted you to draw and label a single DNA nucleotide, and I would just circle the D there because a few people misread it. So your marks here are for the labels and for the shapes. I've drawn the shapes for you, so we should have a pentagon, a circle for the phosphate, and a square for the base. And we need them to be connected in the manner that I've shown in the correct shapes and positions. And then we just need to label those. So there's our phosphate. We know that this is deoxyribose because it said DNA. And then I've written base, but let's make it even better and say nitrogenous base. So that was nicely done. If you're really showing off, you could have labelled that base as A, G, T, or C, so adenine, guanine, thymine, or cytosine. You could not have used uracil because this is asking for DNA nucleotides. So really the only place you're going to go wrong is just if you read this incorrectly. Okay, first real piece of exam technique here. This is a give 2 features and explain. So it's not enough to give the features, you need to tell me how they're important to semi-conservative replication. So there were 3 things you could have said here. You could have said that there are weak H bonds, and they're important because they're easily broken. Okay, remember if these were ionic bonds or covalent bonds, they wouldn't break so easily. That easy breaking allows the molecule to unzip. Okay, second point is that DNA is a double-stranded molecule. That's me giving a feature of DNA, then to explain it, I'm going to say both strands act as a template. And that's important because that's the definition of semi-conservative replication. And the final feature that I'm giving is complementary base pairing, and then my explanation as to why that's important means that it allows accurate replication. Okay, so each time making a statement and explaining why. If you follow those instructions, you can't go wrong, but you have to have both parts to get that single mark. So each one of these points is a single mark on that mark scheme.